Hey guys, so this is lecture number 5. We're starting into Mendel and Punnett squares. So make sure you've got, as usual, a pen or a pencil, piece of paper, notebook, notepad, whatever you need to record these notes on. And here we go. Okay, our objective for this lecture is students will be able to analyze Mendel's work with pea plants in order to describe his impact on modern genetics. Okay, Gregor Mendel. Who was he? He was the father of genetics. He was an Austrian monk. He spent his spare time, which he had a lot of, experimenting with pea plants. And all of Mendel's data really laid the groundwork for modern day genetics. Before Mendel, we really knew very little about how we pass traits on from parent to offspring. Okay? A lot of this lecture is going to be vocab, so bear with me as we go. These first two words we're already very familiar with, but it's just review since it's important to what we'll be talking about in a few minutes. Okay, chromosomes, we already know, are DNA in coiled form. We've known that for quite a few months now. There's our chromosome. And genes, we know that genes are segments of DNA, and they code for one trait. Okay, they occur in pairs. We get one from each parent. So your gene over here on the right, we see is this purple section. Now I want you to notice over here because this purple is going to change color because genes come in different forms. Different forms of genes that appear at the same location we call alleles. Okay, so watch over here as the colors change. This blue block is the same gene. Oops, it's the same gene, but it's in a different form of the gene. Okay, so maybe that purple one codes for brown eyes, and this blue one codes for blue eyes. Okay, some new vocab coming at you: genome, genotype, phenotype, homozygous alleles, and heterozygous alleles. These are big words, but remember this homo here in the front means same, and hetero here in the front means different. Okay, so we're going to start with genome. Definition of genome is all of an organism's genetic material. Okay. Definition of genotype is the genetic makeup or the letters that we assign to certain traits. An example I'm using here is using the letter G. So we use both capital and lowercase letters, and more on that in a little bit. Right now, just get this down. Okay, we're going to say that this person has green eyes, or G codes for green. Okay, so this would say that this person has green eyes. Little g might mean that that's the allele for blue eyes. Okay, but they have a big G and a little g. This is green. That's the genotype. The phenotype is what occurs in the physical makeup. It's what is expressed on the outside. So the genotype for this person looks like this. Meanwhile, the phenotype would just be the color green. Okay. Now, homozygous alleles. Homozygous means the same. So when we have a capital and a lowercase letter, that means for homozygous we have 2 capital letters or we have 2 lowercase letters. And we have the words dominant here and recessive. Recessive being for the little guys and dominant being for the capital letters. Okay. On the other hand, our last word we've got is heterozygous alleles. And heterozygous is when we have a capital letter and a lowercase letter together. It's heterozygous, meaning hetero, meaning different. Okay. So, last few notes on vocab. Okay. I was talking about dominant and recessive. Some alleles are dominant over others. So we can have 2 types of genotypes producing the exact same phenotype. Homozygous dominant and heterozygous both produce green peas. Dominant means to have power over or be stronger than. You could be dominant on the football field, you can be dominant in the swimming pool, you can be dominant on the track. I'm using sports analogies here, but you can really be dominant in the classroom, you can be dominant on stage as a dancer, any place that you're very, very skilled at something. So if you're dominant, you really are stronger and you take over. A dominant allele will appear more often than a recessive allele. For example, in eye color, blue eyes are recessive over brown. Let's see a cartoon that shows us all the vocabulary we just talked about. Okay? Sit back and relax. It's about 3 minutes, and then I'll come back as soon as it's over.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding the pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the two alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers his lowercase friend, so the only time you get green babies is if you have two lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the two heterozygous kids marry, their babies could have any of the three possible genotypes, showing the two possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
Alright, so we're back. So Mendel, out of all his pea experiments, came up with 3 principles. The first one is that there is a dominant allele and a recessive allele. Okay, organisms always inherit 2 copies of each gene, one from each parent. Here are our alleles. And again, I'm showing you where this is where a certain gene if we're talking about flowers, this would be like the color of the flower. Okay, this is where the gene lies on the chromosome, and the different color alleles show that they are separate and they code for 2 different colors. Okay, so one allele is dominant or expressed, while the other is recessive, or usually hidden. The second principle is segregation, and that simply says that when meiosis makes sex cells, each cell has only one allele. The third is called independent assortment. An independent assortment is each trait is passed on independently of other traits. So your eye color is not also attached to your fingernail length. Okay, so just visually, we'll review the vocab we talked about in the beginning of the lecture. Remember, genotype is the letters we assign to different traits. Phenotype is the actual way that a trait is expressed. So in this case, The letters back here are coding for either yellow or green, and the phenotype shows us if it's yellow or green. Okay? Alleles are genes or alternative form of genes that are located at the same place on a chromosome. Homozygous means that you have both two uppercase, or you have two lowercase. And heterozygous is one uppercase letter and one lowercase letter. Okay. Last but not least, Punnett squares. We're going to be doing in class in the next day, 2 days. Punnett square predicts the probability of traits in offspring only if you know the genotype of the parent, the genotype being the letters we assign to the traits. Okay? So as I showed us in the video, this is our Punnett square. Mom would go on the top, or dad, and dad goes on the bottom. Okay? And we put mom's alleles up here, and we put dad's alleles up here. We bring them together, and this is what we would get. So here, big Y is the genotype, and the phenotype for big Y is yellow. Down here, the genotype is little y, and the phenotype for little y is green. So, you're done. Congratulations, you made it. Go ahead and take the quiz, and I will see you in class.